An overabundance of nutrients from agricultural runoff can lead to excessive algal growth and deteriorating water quality. However, this type of pollution is difficult to regulate because its source is not directly measurable. This topic area explores state-of-the-art methods for detecting algal blooms, tools for modeling the origins of non-point source pollution, and regulatory options for addressing the challenge. Our panel is comprised of 4 esteemed scientists and scholars. Dr. Jana Compton is a senior research ecologist with the Environmental Protection Agency's Office of Research and Development. She focuses on identifying the sources and effects of nutrient pollution at local to national scales. Dr. Zachary Easton is the Alphen Professor of Agriculture and Life Sciences at Virginia Tech, where he focuses on water quality in the context of both native and managed systems. The Easton Lab's core research, teaching, and extension programs focus on synergies between soil and water resources, environmental change, biophysics, and sustainable land use in agricultural ecosystems using an interdisciplinary approach. Dr. Kelly Luis is a scientist in the Water and Ecosystems Group at the Jet Propulsion Lab. Her research focuses on the development of aquatic remote sensing algorithms for environmental monitoring, forecasting, and decision-making. And Dr. Bryn Stewart is a postdoctoral scholar in environmental science and engineering at Caltech. She studies the relationship between hydrological and biogeochemical processes in the critical zone and how they influence stream and water chemistry. Thank you so much for being here.
Hi, everyone. I'm Jana Compton. Thank you for coming, or thank you for inviting me, is really what I mean to say. So my father was a physicist, and he got invited to give a talk here in the late 60s, we think. And so this morning I spent my time walking around and finding the statues or the busts of Feynman and Einstein to take pictures and share with my mom. So it's kind of cool to be here on campus, beautiful campus. So I'm going to be speaking about non-point source pollution and the science needs for water quality monitoring. I'm part of the Office of Research and Development with EPA, which is EPA's research arm. So we're more like academics and USGS than we are like the regulatory side of EPA. I always like to say that at the beginning of a talk, especially when I've heard EPA, the Environmental Protection Agency, come up many, many times today. But there were many things that the previous speakers said that resonated with me. I'm going to try to touch on some of those ideas as I go through. I want to acknowledge my co-authors, Robert Sabo, who is in DC with EPA, with the same group at EPA, and he actually was invited to give the talk, and I'm sort of filling in for him because he's got some things going on at home with new babies. And then Amalia Handler, who works on cyanohabs, and then Ann Ray, who leads our nutrient research within the Office of Research and Development. So I'm going to talk about some key research needs. Many of Laura mentioned many of these being issues and concerns. Quantifying non-point source pollution is challenging, but it's really important, and we need to try. We need to do our best. We need to find ways to connect the inputs to water quality impacts. I heard several of the speakers earlier talking about the sources and the endpoints. And then also kind of asking the question of connecting conservation practices, management practices to nutrient loads and water quality information. And so I'm going to go through a couple of things that we work on at EPA, the EPA's National Nutrient Inventory to get at the sources, EPA's National Aquatic Resources Survey to get at the water quality responses, and then talk about some challenges to understand basically the question that a lot of my research is moving toward. I've been with EPA for 25 years, I meant to mention that. Moving from quantifying the sources and the impacts to asking questions about, is it working? Are the billions of dollars that we're spending every year on conservation, on management, on restoring water quality, is it having an impact? Is it doing the things that we want it to do? I think that's for a really important question. So is it working? That's the simple way when we talk to farmers in the Willamette Valley. Is it working? So I'm going to start with the systems diagram. I'm a biogeochemist. We like these box and arrow diagrams more than anything. So we've got the demands, the human demand for nitrogen, just as an example. I have a phosphorus cascade, but I don't have time to go through all this. But the nitrogen cascade developed by Jim Galloway at University of Virginia. We have human needs for energy and food that really drive the movement of nitrogen into the environment. We have a lot of cycling. I guess, I don't know if this pointer works. Oh, there we go. Can you guys see that? Maybe you can, maybe you can't. The cycling on the land, the movement into the atmosphere, and so we have ozone, which is human health and crop health concern, particulate matter, same for human health, and lots of other impacts, greenhouse gas effects, stratospheric ozone depletion. Actually, N2O is the largest stratospheric ozone depleting chemical that there is right now. We also have movement of that nitrogen into water. And the concept of the cascade is really that one molecule can move around and have multiple impacts, and that's why we have the cascade. And then we have the endpoint. So we have these externalities. Nitrogen moves beyond its intended use and moves into other places where it can cause drinking water contamination. We've done a lot of work with nitrate in drinking water across the country and also locally. I didn't put any slides, but I'd be happy to talk about that at some point. The health effects of smog, coastal hypoxia, and also cyano-harmful algal blooms, and so cyanoHABs. And so all of these endpoints, very important, and kind of making these connections is very important for the scientists. So I'm going to speak a little bit about the EPA's National Nutrient Inventory, which is how we determine where nitrogen is coming from and where phosphorus is coming from in the environment. So I made I like these Sankey diagrams. I had an engineering student work with me, and so I'm getting some head nods. These are engineers, a lot of engineers in the room, and the Sankey diagram has been really helpful for looking at the fate of nitrogen and phosphorus. You can see ag fertilizer, farm fertilizer is the largest source of nitrogen if you just look at the conterminous US, largest anthropogenic source of nitrogen to the landscape. The second largest source, because we have the Clean Air Act and we cleaned up a lot of our nitrogen deposition, the second largest source is now crop biological infixation, things like soybeans and alfalfa. And then total deposition is the next largest source, and then we have things like livestock waste, natural biological infixation, which is not necessarily anthropogenic, and then other smaller sources. All of these sources can be important depending on where you are in space and across the landscape. You can see some of that nitrogen goes into crops and some of it stays in the environment as what we call surplus, goes into soils, may go back up into the atmosphere, it may leach into groundwater or run off into surface waters. So in the inventory, I just put a little QR code if anybody wants to learn more about it and see some of the papers. What's in the inventory, we can track anthropogenic inputs and outputs. We can track pollution through time. We can identify the largest or dominant source. We can communicate with people. And we can explain patterns in water quality over time and space. We also put together a phosphorus budget. Phosphorus looks really different. It's not as complex as nitrogen. There's not as many atmospheric forms that move around and do things. But so you can see here, actually, the largest source is livestock waste. The largest source of inputs to anthropogenic inputs to watersheds across the US is livestock waste. I just put these figures together a little while ago, and it was kind of surprising to me within the last year that that's the largest source for phosphorus. It's kind of different. It's different than the nitrogen inputs. Then we have farm fertilizer, obviously, is a very large source and nearly as large as the livestock waste. And then we have another host of inputs, including human waste, which is important depending on where you are. So I'll talk about the spatial patterns. I have a lot of maps of the US in this talk. So in the center of the US, we have fertilizer in yellow in farmland. And other parts of the country, I'm from Oregon, so I kind of go to the northwest, but we have many parts of the country where fertilizer is the largest source. This is a HUC 8, Hydrologic Unit Code 8, which is kind of a nice scale to look at the US watershed landscape. So this is also livestock waste. You can see kind of on the edges of farmland, and especially in the southeast, livestock waste is the largest source of nitrogen. In red is human waste. That's near cities. For example, Puget Sound. In Seattle, there's about 65% of the nitrogen going into Puget Sound is from human waste. And then the blue is deposition. So that's where atmospheric deposition, that's the largest source in both the east and the west. And so you can kind of see these different spots on the landscape. Phosphorus, there's a lot of similarities. You can see where farm fertilizer in the center of the country is the largest source. But you can also see this importance of livestock waste, particularly in rangeland in the west, and just other areas all throughout the southeast, very important. And so we have some areas where deposition's important. We have a few areas where human waste is important, especially along the East Coast. So in terms of connecting the inputs to the water quality endpoints, we have the Clean Air Act and the Clean Water Act, and there's a number of things that, there's scientific requirements and scientific questions that are associated with these mandates from Congress that are associated with these acts. But the administrators of EPA, for example, the first administrator, and I think maybe the fourth or fifth, William Ruckelshaus, he said, "What do you mean you don't know how many acid lakes there are?" So that was a question to the science community within, the broader science community, but particularly within EPA. A congressman from Illinois, are our waters getting better or worse? William Riley, who was the administrator under Reagan, said, "Good news, based on my years in the environmental movement, I think the agency does an exemplary job of protecting the nation's public health and quality of the environment." I think a lot of what was said about the drinking water systems, and I think that is true for many, many chemicals. Bad news, I can't prove it. So they didn't have the data to really address these questions. And so in response to that, in the early 2000s, the agency, and particularly the Office of Research and Development, developed the National Aquatic Resource Surveys, and they survey 4 types of systems: rivers and streams, lakes, coastal condition, and wetland condition. And so these are every 5 years, these surveys are conducted. I'm going to talk a little bit about the National Rivers and Streams Assessment, mostly about the National Rivers and Streams Assessment, and a little bit about the National Lakes Assessment. These surveys are designed to sample all of the U.S. flowing waters. It's 2,000 samples about every 5 years, and so I'm going to present data from three surveys, and it's about 5,000 samples. It's basically sending a crew out to measure all kinds of stuff, and so they go out for one day, and they in the kind of the growing season, and they measure physical habitat, stream flow, insects, fish, algae, water chemistry, which is what I'll be speaking about. They use a probability-based design, so we can say something about ecoregional status and also national condition of the waters. This probability design from our main statistician allows weighted estimates of the entire coterminous U.S., which is about 1.4 million miles of streams. So we can take those numbers and we can translate them and extrapolate to that whole stream miles. And the recent report came out in 2018, from the 2018 to 2019 data came out last winter. So for each of the little dots on the map, that's a stream sample. So we have about 5,000 samples between 2000 and 2014. And each one of those dots, we can draw a watershed upstream, and we can say these are the largest sources. So we just take all the sources and we look at the largest ones. So we can kind of simplify things and look at what's important. And it looks kind of like those maps that I showed you earlier, but maybe a little bit different, a little more detailed. Obviously in the center, on the left is nitrogen and on the right is phosphorus. In the center of the US, in the Corn Belt, we see farm fertilizer as the largest source. Along the edges and in the southeast and many areas of the west, we have the brown, which is livestock waste, as the largest source. We can see areas in the west and in the east and the upper Midwest where we have deposition as the largest source. These are largely forested areas or natural areas, or areas where there's multiple sources kind of coming together, but deposition is the largest one. And then you can see in the both maps, there's kind of an orange color on the phosphorus map, which is human waste. There's a kind of pink color, which is the urban fertilizer, that's human waste. And we also can track urban fertilizer, which is non-farm fertilizer, things like golf courses and yards. And so I guess one important point is that there are some large sources, but there's also variation across space, and that's something that tells, we can tell something about what the management needs are across space. So the proportion of the US, this is taking that approach using the statistics to be able to say what the national pattern is. And so across the US, for the stream miles, across the flowing stream miles across the coterminous US, for nitrogen, about half of the streams, agricultural sources are the largest, and the deposition for the other half. It's very different for phosphorus. For phosphorus, about 60% is agricultural sources, closer to 70% is agricultural sources, with manure being the largest source. We can also zoom in and look at sites which have poor nutrient condition. So either poor for nitrogen on the left or phosphorus on the right. And you can see for nitrogen, we kind of increase the share of agricultural sources. So agricultural sources are much more important in areas where we have poor nutrient condition. For phosphorus, it doesn't really change that much. It shifts a little bit to increase, but we clearly have a lot of phosphorus sites, which are where ag source is very important, and other sites, where waste is important or deposition. And this is just the summary for each of the, for both of the surveys, for the 3 survey periods, most recent survey periods, 8-9, 13-14, and 18-19. Nitrogen on the left, 44% of streams across the U.S., stream miles across the U.S. are considered to be in poor condition for nitrogen. And then for phosphorus, it's gone from 51 to 59 to 42. It's varied over time. It looks like there might be an improvement in phosphorus, but it's not exactly clear what's driving that. But certainly we're seeing very high proportion of streams that are in poor condition with respect to phosphorus across the U.S. I'm also going to speak a little bit about using the data to look at some of the progress associated with the Clean Air Act. This really great movie shows you total sulfur deposition over time, and when it starts red, that's 2000, and then sulfur deposition has declined dramatically, about by 70% over the last 20 years or so because of the Clean Air Act amendment. So you see this improvement in air quality, especially in the East, where we had a lot of coal-fired plants. We look to see, is there a change in water quality associated with this Clean Air Act amendment? And so we do see nitrogen deposition has declined. That's kind of what the top graph shows you, with the red changing to pink or blue, especially in the East. Nitrogen deposition has declined. But what we do see is that stream organic nitrogen and stream organic carbon has increased over time. We're not seeing a difference in nitrate, and we're seeing an increase in total organic nitrogen and carbon. Not sure exactly what the drivers are, but it has implications for disinfection byproducts across the country. And it's an important but not very well studied aspect of what's going on. We don't really know what the mechanisms are. We have some hypotheses, but we need to better understand that. We've also been connecting the source information to cyanohabs, and so this is just the QR code for the Cyan Project, which Kate spoke about. This looks at the 2,200 largest lakes in the country. So cyanohabs can be a problem in small lakes, and there are better satellite data. Cyanohabs can also be a problem in benthic zones in streams and rivers, and that's something that we're just starting to learn about, just starting to study well, so that we can look at that. But we know that nutrients are a key driver of blooms. We're taking our inventory data and we're looking at the National Lakes Assessment information on, for example, the toxin microcystin, which is a widespread toxin associated with cyanohabs that's often monitored by drinking water utilities. And so this is one that we are trying to understand what are the drivers. And you can sort of see that there's some hot spots, but there's also places where you might have a clean lake right next to a lake that has high microcystin. And so there's just a lot of things that a lot of unknowns and uncertainties behind this risk. This is a probability of risk of microcystin. So on the last thing, I don't have a whole lot of time. I'm going to try to go through, is it working? And go quickly through that. And so we do a lot in terms of conservation practices. This is the USDA's Conservation Effectiveness Assessment Program, or CEAP, which is comparing 2010 to 2020. We see we've done a good job in reducing surface runoff over the last 10 or 15 years, but subsurface leaching continues to increase, and that's actually the largest, especially for nitrogen, that's the largest source of nitrogen that's moving into the landscape. And so maybe we need to do a better job. The implications would be we need to do a better job in reducing that subsurface leaching, especially when we're thinking about groundwater. So I'm just going to try to highlight some key research opportunities quickly here. Measuring nutrients related to the environment. We need maybe better information about how do we map and monitor fertilizers and manure. Phosphorus deposition, we're trying to get a better handle on it. It's not actually part of the NADP or the National Atmospheric Deposition Program, but they're trying to develop methods to improve those measurements because it does seem to be important in some places. Tracking crop harvest and surplus for management relevance, also soil testing, adding that. And there's a lot going on with animal systems and nutrient management associated with animal systems that we need to better understand. Water quality information, I think we need more machine learning approaches, which others have spoken about. And I really agree with the idea of all these young people coming in with strong R skills and coding skills and are able to make R shinies for the state. We've helped that at the state of Oregon that can create story maps that are used by the state or local entities. It really, there's a lot of folks with a lot of experience that are retiring, but there's a lot of young people with new, different skills that are coming into these jobs. Better access and analysis of existing water quality data. I would say the water quality portal, there's a lot of data there. There's also a lot of data collected by drinking water providers that I've rarely seen scientific studies that have looked at that data and presented it and tried to tie it to condition over time. It's some of the best data in the country that we have that the drinking water providers are collecting on the raw water or the source water. We need to look at that data and try to see what's going on and maybe put different systems together. Identifying gaps, there's a great Duke study that looks at this. If you just Google Duke and water quality portal, you'll find it. I have the link in the top. Cheaper, better sensors, use of satellite data. We've already heard all about that today. And then I think one of the things that I'm really on a bandwagon about, or whatever the word is, connecting conservation and management efforts to water quality, making ththose connections, doing a better job of making those connections. We have all this money, the infrastructure law, the BIL, all this information, these projects are going out. Are we seeing the changes in water quality that we're expecting? I think we're not closing the loop there, and I think that's one of our big challenges, and I'll stop there and be happy to, I guess at the end we'll have questions. So thank you very much for the invitation.
I'm going to kind of tell a cautionary tale, I guess, about relying too heavily on models to drive non-point source programs. And this is basically stems from a major report we did for the Chesapeake Bay TMDL. So there's a TMDL in place in the Chesapeake Bay that's the largest TMDL ever implemented. And I've been part of the EPA Chesapeake Bay Scientific and Technical Advisory Committee for the past 10 years as a body of approximately 40 different academic and government scientists that try to guide the EPA Chesapeake Bay program on water quality improvement practices. And so we did this major report. It was released last year, May of last year, trying to identify some of the response and implementation gaps that we're seeing in the Chesapeake Bay program with respect to water quality, and particularly non-point source. We realized very early on that really was a non-point source contribution to water quality pollution that was really the big problem. And so I'll focus on that here. So the motivation for this report was simple. Since 1985, there's been mandates, and then in 2010, the TMDL for the Chesapeake Bay estuary to improve its water quality. And so this is a time series of the percent water quality standards attainment in the Chesapeake Bay estuary, 1985 to 2020. And what you see here is that we've seen really very little water quality improvement over this 40-year period, nearly 40-year period. Trying to reach 100% attainment in the Bay, we're seeing somewhere between 25 and 30% attainment on any given year. And we wanted to know why, or look into why that might be the case. And so again, very, very early on, we realized that it was the non-point source pollution that was the major problem. The wastewater treatment plants have all upgraded to tertiary treatment. Their loads have declined dramatically in the past 25 years, whereas the agricultural mainly, but also urban non-point sources are a much larger proportion of the total load to the Bay. And so one of the major findings was that with respect to the TMDL and non-point sources was that we're not really seeing enough adoption or behavioral change in the watershed. We termed that the implementation gap. So there just aren't enough BMPs on the landscape to meet the water quality goals that we've set out for in the TMDL. The other one was that these practices may not be as effective as this model, which I'll talk about in a minute, predict they are, and this is the response gap. So BMPs aren't working like we think they're working. But before I talk about sort of the model, I want to briefly go through the system. I'm not going to go through all the linkages here, but really the non-point source implementation policy In the Chesapeake TMDL is really structured into 3 sort of tiers. Is the policy component, so the federal permitting, state non-point source programs, and then the accounting system, which I'll talk about in a minute. And that gets implemented. We're trying to change these behavior in the watershed, so behavior of water quality managers, producers and land managers, and service providers as well. And all that happens before you get down here to this physical response. So this is the physical response of the watershed. What sort of loads are we seeing leaving the watershed? And again, pretty complex. There's linkages among all these, behavioral components, the policy component, and the physical response. And I guess just to point out here, it's a complex system. Lots of linkages, lots going on. There's a lot going on before we ever reach, BMPs being installed, best management practices, I should say. So these are the practices we install on the landscape to, we believe, try to improve water quality. But there's a lot going on here before we ever talk about implementing BMPs, and that largely gets ignored in the program for the Bay. I guess I'd also point out that there's a lot of uncertainty. All these linkages and levels have a great amount of uncertainty associated with them. And there's tremendous heterogeneity and diversity, again, across all levels and linkages in this system. So what does the TMDL rely on to track, progress in meeting the TMDL? Well, it relies on this model called CAS, which is the Chesapeake Assessment Scenario Tool. It's just a basically empirical model. Uses lots of export coefficients to predict the average nitrogen phosphorus and sediment reductions from BMPs, and it does it at a really coarse scale. So 20,000 acres is the finest sort of unit it predicts at. A lot of them are quite a bit larger based on geomorphic and land use conditions. And the point here is that this model makes a lot of simplifying assumptions about how the watershed operates and how non-point sources are integrated into that watershed. Really, it just takes nutrient input sources like fertilizer, manure, and atmospheric deposition, multiplies them by some export coefficients and estimates of runoff losses to get in first case, the average NMP sediment losses from that land unit. And then when BMPs are applied, the average reductions from BMPs. And the point here is that all the decisions that the states and the water quality programs make are based on estimates from this CAST model, right? So there's no real empirical data. There's not a whole lot of measurement that underpins this. Yes, the model is to some extent calibrated to, major river monitoring stations, but it makes a lot of simplifying assumptions. And so really this model is what drives implementation of BMPs across the landscape. And so the standard water quality program, particularly agricultural BMP program, what they do is they offer a part of the cost to install a BMP, 40% to install a manure storage lagoon or something like that. They don't cover 100% of the cost share. And that's intended to drive adoption by, let's say, producers in the watershed who adopt that BMP across the landscape. And then what the program does is assume that since that BMP is installed, it's, say, reducing phosphorus loss by 30%. So the state reports the number and types of BMPs using that sort of removal efficiency and the acres treated. That gets put in the model, and the model says, okay, well, you've reduced phosphorus loss by 20% across that the area that BMP covers. The problem with this is that, first of all, producers aren't usually going to adopt BMPs that don't improve their production practices. So in this program, you see a lot of BMPs being cover crops and nutrient management plans. You don't see a lot of innovative BMPs that might be quite effective at reducing nutrient loads because they're too expensive and they don't improve the bottom line of producers. And so what does this lead to in terms of an actual water quality response? One of the questions we asked was, is the watershed responding to management as expected? And so on the top here, I'm showing predictions from this CAST model, again, simple export coefficient model for total nitrogen and total phosphorus for these different sources in the watershed. And I'm going to focus on the non-point sources here, so crop, pasture, and urban non-point source developed there in gray. And so what the CAST model tells people, and again, this is the model that drives implementation of BMPs in this watershed, is that, maybe it doesn't, is that For nitrogen, we're seeing some moderate reductions from 1992 through 2012. So a moderate reduction in nitrogen. And for phosphorus, it's actually a little bit more significant. So quite a significant reduction in total phosphorus losses across that non-point source sector over that 20 or so year period. The problem is when you compare that to empirical data or measured data, so this is empirical estimates using the Sparrow model for TN and TP, we see quite a different trend. So for nitrogen, you could probably argue it looks somewhat similar. So total load delivered to the estuary declined a little bit based on cast, pretty much no change based on Sparrow. But the real discrepancy occurs for phosphorus, where the empirical or the measured data are telling us something quite different than what the water quality program policy model is telling us. In fact, it's telling us that total phosphorus losses have increased quite substantially over that 20-year period. And so we wanted, well, that was all there. So this is just another example. This is a slide I used on Wednesday at a symposium in Virginia, looking at non-point source programs in Virginia and how they're functioning to improve water quality. And so this is a figure showing 3 different time series. 2 are that cast model here, just different versions of it. And 1 is the empirical estimates. So this is basically WRTDS, so watershed regression on time, discharge, and season. So it basically normalizes the flow, takes out some of the interannual variability. And we're looking at the phosphorus percent of 1995 load here. So 1995 was the base year that the TMDL is using to estimate water quality improvement from. And so you can see here that if you look at the estimates from that cast model, that's the WIP target as well. So that's the goal we're trying to reach there. We've got to reduce our phosphorus losses by about 40% from 1995. You can see that the cast model says we're there, right? We've made our goal. If you look at the measured data or the empirical data, it suggests quite the opposite. Again, quite substantial increase in total phosphorus losses in this, 3,000 square mile or so watershed here. And so I want to talk a little bit about some of the things we identified as opportunities to improve non-point source programs. Because like I said, this program relies almost entirely on that one model to drive all of the implementation in the watershed. So something we thought quite a lot about in this report was what are the opportunities to improve non-point source program performance. And one of the things we arrived at quite early on was the nutrient mass balance problem. This was talked about a little bit in the nutrient inventory in the previous slide. It's a little bit different terminology here. I'm showing an example basically based on a farm, right? So the nutrient mass balance basically describes the inputs and the outputs and the change in storage of any constituent. In this case, we're going to talk about nutrients. Oops, sorry. I need to stop using the animation. It never works. So for nutrient imports, you have imports of feed, fertilizer, maybe bedding and manure, could be animal numbers here as well. Those are importing nutrients into this farm system. Could be a watershed, it could be any system you're looking at. And there are nutrient outports, so products and crops, animals, manure. I think I had biomass down here as well earlier. And so what you want to try to do is balance those two so that you don't get this surplus occurring. So in a lot of the watersheds in the Chesapeake Bay, we see a pretty significant surplus of particularly phosphorus in those heavy livestock regions. So what happens when you get that surplus is you get the buildup of those nutrients in the soils or groundwater or biota. And those also lead to increased exports in water and for nitrogen atmospheric losses. So we thought this was something where better data and monitoring of particularly farm level mass balances could go a long way to improving the program outcomes. And this is just an example of something like that. So this is a paper that was just released, Jimmy Weber at the USGS, looking at 3 different showcase watersheds in the Chesapeake Bay region, where the states and NRCFs have spent upwards of $250 million in these 10 square mile watersheds to improve water quality. And what they found was that over the past 3 decades, we've seen a nearly 400% increase in the number of BMPs installed in this watershed. So this is actually a stream I work in extensively. We're showing a riparian buffer here. Everything looks like it's being done right down by the stream. The problem is that they've also seen a pretty significant increase in the number of animal units in this watershed. So they more than doubled, right, up here, those animal units in that watershed. Can anyone guess what the water quality trend might look like? It's actually increasing. So both TN and TP are increasing quite substantially, particularly for TP, due to the result of that, increasing in that mass balance. We also found that nutrient loads, they not only vary across space and time, they also vary across land managers. So this is a study actually in the Shenandoah Valley, a heavy livestock area of the Chesapeake Bay region, looking at the phosphorus mass balance across 58 dairy farms. Again, these farms are all located within 30 or so miles of each other, so similar geographic region, similar farm types, but you see a huge range in those phosphorus mass balances from farms that are, basically storing phosphorus. They're not releasing the phosphorus in the environment, to farms with 100 kilogram per hectare mass imbalance. So there's 100 more kilograms per hectare coming to that farm than is leaving in crops or protein. And so we don't really know anything about what the distribution of, say, phosphorus or nutrient mass balances might look like across the watershed. And these are just two kind of example distributions of what different farm types might look like. Again, this is a place where more monitoring and analysis of data could go a long way to improving our understanding and management of the system. And then finally, one last example would be looking at sort of spatial targeting of nutrient hotspots across the landscape. So the way the CAST model works would be represented by this figure on the right here. So we're using basically average conditions under huge areas. So, you really can't kind of distill down to a field level and determine what's going on. And so we did some analysis on looking at what the phosphorus loss might look like under different or under site-specific conditions. And so you can see here that under site-specific conditions, there's relatively small areas of landscape that are these hot spots of total phosphorus loss. And so, would you manage this landscape any differently if you assumed one versus the other? I think you probably would. And then finally, some of the innovation that we had talked about or proposed in this report dealt with trying to incorporate outcomes into the kind of loop here, right? So down here in the bottom left is how CAST operates now. So CAST drives the implementation programs, producers adopt, there's a BMP, they report it back. Well, what if we thought about it a little bit differently? So what if instead of BMPs being the metric we were trying to reach, what if it was outcomes? And that could be any number of things. It could be, a decrease in soil test phosphorus, or a decrease in the nitrogen mass balance, or an actual ambient water quality measurement. And if we focused on those, could we improve those outcomes? And that would necessarily need to include additional monitoring and analysis, either or finer scale modelings. And the other sort of component of this would be to pay for actual outcomes, right? You're not paying for a practice to be installed. You're paying for a reduction in phosphorus loss from this watershed or some other outcome. And so we thought that might be a little bit different way to think about, how this program might evolve to improve water quality. And again, this is just another example of that. So if we want to incentivize outcomes, we can think about it along a spectrum. So you've got these three BMPs, cover crops, exclusionary fencing, and a bioreactor. And there's sort of a continuum of installation costs and benefits, right? Cover crops are low cost. They have private producer benefits. That's why they get adopted readily. You have these bioreactors, which has high upfront installation costs, doesn't provide a producer with any benefits. Why would they adopt that? But under voluntary cost share programs, right, which is what most of the agricultural BMPs are, adoption rates fall from left to right. So that's why cover crops are so widely adopted. They improve production practices. And then I could sort of have two questions with this. So which is the most cost effective? And maybe that's sort of just a general question. Cover crops are cost effective for producer, but probably don't provide a whole lot of environmental benefit. Whereas bioreactors might be very effective at reducing nitrogen leaving a field, but they don't provide any private benefit? And then which practices provide the most assurances of delivered reductions? Again, it's very hard to go out and quantify how much nitrogen a cover crop is actually prevented from leaving a watershed, whereas something with a pipe in and a pipe out, you could perhaps measure much more easily. Again, another opportunity to incorporate enhanced measurement into the system. So with that, that's all I have.
All right. Aloha mai kākou o Kelly Mariko anoio kupu'uwai Lewis ko inoa no wai ehu ma kamokopuni o Maui he kanaka noi I pekema ma NASA JPL. Aloha everyone. My name is Kelly Lewis. I'm born and raised in Waiahu, Maui. And I'm, as of this week, was appointed to a scientist position at NASA JPL. I intentionally introduced myself in my language because I think my island and where I grew up was probably my greatest teacher in how non-point source pollution impacts people on a day-to-day. I live maybe about 10 minutes from a river, and whenever it would rain, it's cloudy, and as kids, we want to go swimming in the river like right after. We're like, we didn't get a chance to go play outside. But my mom, or just our parents in general, would be like, all the wild boars that run in the mountain, that rain, what is it going to do to our river? It's going to bring in potentially their waste in the river and bacteria. There's a ton of cesspools in Hawaii that says 83,000 cesspools. It's the most of any state. Those cesspools in people's yard. If there's an overflow with the big rain event, where might that go? And we're like, in the river. So we'd have to wait. And I kind of share this story with you because I think from a remote sensing point of view, there's really an exciting future where we have a lot more sensors and instruments and data sets coming about. But I think one of the really novel parts of what's going to happen is when we think about relating these awesome new data sets to people's actual lived experiences, that will do a way better job at integrating this type of incredible Earth observations information at a much quicker pace for decision making. So I kind of wanted to kick this off by sharing an image actually that my intern, her name is Jana McFarland, that she shared with me of the Upper Klamath Lake, let's see, right there. And here she is. So this is Jana McFarland. She's from Northwest Indian College, Squaxin and Skokomish in Washington. And she shared with me about these, wrong one. These endemic fish species. They're really cute, these sucker fish, and they lived in this lake. And that this is a neighboring tribe to hers, and she wanted to study Upper Klamath Lake because there's an EMIT image. So that's EMIT is the Earth Mineral Dust Source Investigation spectrometer that's on the International Space Station. In my new role, I was recently appointed to be the aquatic applications lead for it. So we've been working on trying to see aquatic applications of this new instrument on the ISS. So we were just like, all right, let's take a look at what you're seeing. And I think over the time she was starting to teach me about like this is one of the largest lakes in Oregon. It has HABs every year. It has changed the way people interact with that lake. So what she did was she, as mentioned, looked at the cyan imagery that's from Sentinel-3 Oceanland Color Imager, which is at 300 meters, and started to compare it with the EMIT imagery from the International Space Station. EMIT is at a finer spatial resolution. 300 meters for Sentinel-3 Olchi versus EMIT, which is at 60 meters. I know 300 meters versus 60 meters is not really a good matchup comparison, but a 60 meters width is about roughly the width of a soccer pitch or football pitch. So when we start to look at this imagery, you see much finer spatial patterns, which really started to push you to think, why are we seeing that there? Are there certain places around the lake that is actually more conducive to creating certain patches within the lake? So Chanel has since gone on to, now she does astrobiology research, applying imaging spectrometers to other planets. But she kind of started to help me repackage how do we think about remote sensing of non-point source pollution. And I kind of made a cute 3 C's of different ways we think about it. It's mainly a memory tool for me to kind of encapsulate all of the remote sensing research that's done at JPL and at this axis. So I kind of think about it, we can look at things like surface cover. So this is an EMIT image over North Africa, and one of their key objectives was to map key minerals that, if lofted into the atmosphere, has essentially cooling or warming impacts essentially on our planet. You can also take that to further not only map minerals, but vegetation, aquatic ecosystems like Chanel, but as well as starting to look at the composition of shallow benthic ecosystems from a more of a catalyst perspective. What are those big mechanisms that move, whether it's nutrients or surface cover, and changes it in a way that ends up in water bodies? We have a lot of people that work at the wildfire front as well as floods. Not exactly an expert on the floods, but I'm happy to connect you with people in our group that work on the SWOT instrument, which are looking at that. And then lastly, we're looking at consequences and Lake Erie Modus Aqua image. And this is where we can start to see, all right, when we have our surface cover, we have a sense of adjacent things going on. We can study maybe these catalyst events that change that cover or change what might be input into a watershed or systems. And then we get to this, all right, what type of algal bloom might it be? Or is there a situation where we might need to really think about our public health advisories? So I'm going to touch upon these really quick. So I also have a different hat. I don't really wear a lot of hats, so I've been thinking about changing it to like shoes. Like I wear a lot of different shoes because I have a lot of shoes. But I sit as a NASA Surface Biology Geology Mission's aquatic coordinator for the Visible to Shortwave Infrared Instrument. That's a mission that's going to be launched later in the decade. That's going to be mapping land cover composition. And right now we're actively developing algorithms that can start to look at shallow ecosystems, piece together get information on bathymetry, the water column, optical properties, how that might relate to biogeochemical properties, as well as benthic fractional cover, and those suite of those things together give us a much better understanding of those conditions there. And selfishly, I obviously am starting with the Kaneohe image. Also, there's really burgeoning work that's led by Michelle Gurak, principal scientist, and actually starting to look at potential plastic sources, not only with the MIT, but with airborne imagery, specifically Avarice Next Generation. And this is a map created by Phil Broderick, also at JPL. And he's applied a plastics matched filter approach over this area in India, and started, when you see those like blue speckles and yellow dots, you're looking at really specific features in the shortwave infrared. And if there's like an absorption feature there, that might be indicative of a plastic source. So we're just starting up a lot of this work now. On the catalyst front, this is led by Mandy Lopez these days, and she's looking at coastal watershed linkages between land and sea in relation to wildfires. And when you're combining that remote sensing and modeling information, you can get more information about what is the discharge rate, sediment loads, bacterial information. Feel free to contact her, but I'm also more than happy to make connections. She's working a lot for Woolsey fire impacts. Lastly, Christine Lee. She's a group supervisor of water and ecosystems and do a lot of partnering with UC Merced's Erin Hester. And this is a recent paper that her graduate student, Brittany Lopez Barreto, published in San Reservoir, and she looked at Sentinel imagery, so Sentinel 2 and 3. So those are both multispectral sensors from the European Space Agency, and they're getting a chlorophyll algorithm and applying it across of the reservoir, and then comparing that with toxin information here. And one of the big things is that when you start to piece together all of these Earth observations, you can start to contextualize in scenarios where you might not have somebody out on a boat taking a water sample. So I just wanted to do a brief highlight on all of the great work being conducted in our group. And I'm kind of going to transition now into really some of the key challenges that are coming up, and really in relation to a lot that has been coming up in this workshop. So a lot of my training has been actually on the East Coast, working with citizen scientists, water quality groups. And I don't know how many of you have spent time on the East Coast. It's pretty cloudy. It's cold, so to go and get a sample in the middle of February is not fun. There aren't a lot of volunteers. So started to do a lot of that work. And, a lot of the big questions come up is, well, we can work with individual sensors and satellites, but how do we synthesize them to actually fill in gaps in information? And this is a really nice visualization made by the Goddard's Scientific Visualization Studio that's showing that there's really a rich history of heritage and operational ocean color sensors, or these sensors that can look at making maps like chlorophyll and relating them to things like cyanohabs. And how we go about knitting together from the 1970s to present, that's really an open question and thing we really need to start thinking about. But also one of the most exciting things about my career at this stage is that those yellow sensors, those are new types of imaging spectrometers. So in the past we focused a lot on multispectral ones, and now we're going into imaging spectroscopy or hyperspectral sensors. So I'm gonna give you a visual, is that, so visible light wavelength range, the ones we can see with our eye, rainbow, since early 2002 for global current missions, these are really snapshots across the visible light wavelength spectrum. And they're positioned and designed to coincide with specific water quality features that would pop up that can then indicate that can be useful for a water quality map. However, we're going into new instrument designs that actually have way more finer spectral information. And one of the ways that I've heard the PACE satellite that actually just launched in February, their first light imagery just came out yesterday. I should have thrown that in here. One of the ways they kind of explained this is, imagine for the past 20 years, we've been drawing the color of the ocean with a box of 8 crayons. And now, as of yesterday, I've given you 100 crayons to color the surfaces, like ocean color, or the water surfaces. How is that gonna change the way we look at water quality? How's it gonna change the process in which we do it? And that one. And this is really a visual. So if you were to look at VIIRS, you would have, 5 different points along the visible wavelength range. It's very difficult to determine which one might be a harmful algal bloom, what might not be. But when you have more of that spectral information, you can then start to more concretely relate whether it might be more similar to a HAB species that somebody experiences versus a background diatom spectral feature. So this is actually from Indian Ocean Noctiluca blooms versus that background diatom. In addition to that, we're moving into the space where more of these are being built across space and time, and we're gonna have these incredible spectral information that not only are we starting to look at really specific features like phycocyanin, pigments, community composition, but that will be also in relation to CDOMs, descended solids. And in many ways, I think there's so much promise in all of this information. But wow, there's gonna be really so much that we really need to winnow down and filter to make it reasonable and accessible to people. So some of the work I did early in my postdoc was just focusing on 2 sensors over Corona brevis blooms, so red tides, and along central Florida. And then you can see the red tide. It shows up in ocean color imagery, but also you can see it there. And I think one of the most surprising things for me was that there are these weekly maps that have been generated since, I guess, the late 2000s. And you see that there you can see the cell count along the coast, and that background map is actually modus aqua, or an image of fluorescence. And I was just like, I found this on Facebook. Like, this is on US News. This is, the public is exposed to some of these types of measurements. And it's just like, why not? I think when we were talking, the earlier panel, it's just like, let's lean into how do you make measurements or instrument design that's complementary to this type of information. And one of the really cool things of working with Christian Frankenberg here at Caltech was he was starting to put out these papers related to Sentinel-5 precursors TROPOMI instrument, tropospheric monitoring instrument. And actually this instrument can detect fluorescence in variable atmospheric conditions. So what we did was, we're like, well, maybe we can fill in some of these gaps on a weekly basis. So we wanted to check first if they were consistent. And actually there's a really big bloom in the 2017 through 2018 time window. And we found there's like, wow, so during these really big blooms, we have these two sensors that are complementary, they are agreeing with each other. And then we wanted to look at, all right, let's look at a week where they said there's this enormous bloom that happens in the weekly composite. And then when we pulled the map, we realized for the Modus Aqua, or this imaging spectrometer that can really only detect in like atmospherically clear conditions, we only really had one image from that like really big week. But when we start to find these complementary sensors, we can start to see how they can fill in gaps for one another. So this is some work that we're trying to continue to grow now, not only tying in things like Modus Aqua and Tropomi, but other currently operational sensors like Sentinel-3, Sentinel-2, et cetera. And timing-wise, I might have to run through some of this, and that's fine. To kind of go back to emit at those finer spatial scales, one of the questions when we were working on this project with Chanel was also can something in a finer spatial resolution inform downscaling these larger pixel sizes? I mean, and this was just timing worked out. So we're just starting to dive into this now. And what we're finding is that if you were to take a 60 meter pixel and then you would take a bigger, or take a 3x3 meter or 3x3 box, average it, and kind of keep going up, you would find that a lot of the there you are. We utilize a lot of the spectral information in the 620, 650 region. So we see as we get bigger, spectral information or spatial information, these properties are actually relatively consistent. It's actually once you get to the farther end of the spectrum where you have actually more sediment interactions and all of that. So another really open question we're working on, as well as optical and thermal properties. On the International Space Station, there's also EcoStress, which is a thermal infrared radiometer. So now we can start to look at what is that relationship between cyanobacteria, I say expression, at least, I don't know, I think sometimes at these snapshots, you're like, what part of the life cycle are you in? Like, we still need to really dial in on understanding their expression at the moment a sensor goes over them. But when you can start to pair that with something that's mechanistic, like a thermal property, you can start to piece together, are you seeing more HABs in warmer waters? What does that mean for increased stratification in these waters that might have experienced incredible warming in advance? So I think to kind of take us to the end, just a general recap with remote sensing instrumentation. If it's helpful for you, I think about it in a cover, catalyst, and consequence sense. We can start to look at really the surface color or cover. Really, what are these agricultural fields, compose them from imaging spectroscopy, starting to look at remote sensing during catalyst events where that might move surface composition into waterways, and then potential consequences like what you're seeing in this HAB here in Upper Klamath. And given the big challenge of lots of new and exciting novel instrumentation, I think really one of the most exciting parts of my career is thinking about how do we co-produce new data sets and products that get us quicker to decisions. And yeah, I guess very happy to be here and collaborate with folks that are interested. Thank you.
Great. Hi, everyone. My name is Bryn Stewart, and I'm a postdoc here at Caltech. And today I'm going to discuss some of my work on connecting subsurface processes, stream water quality, and climate change, and how all of these things are related to non-point source pollution. So I study water quality at the catchment or watershed scale, where catchments or watersheds are just areas of land that drain all inputs of water to a common outlet, as shown in this figure on the right. And the reason that we study water quality at this scale is because catchments provide a boundary and a specific set of conditions for an ecosystem. So everything from the climate of an area to the vegetation on the land, the soils, geology, et cetera, all of those different characteristics influence the different reactions that occur within our catchment and sources of different solutes and how they kind of affect our stream water quality. And so water quality and non-point source pollution are driven by these landscape characteristics and catchment processes. And so as an example here, based on the topic that we're focused on today, we know that harmful algal blooms are largely caused by excess nutrient pollution. And so typically coming from nutrients like nitrogen and phosphorus, and those nutrients need to come from somewhere on the land. And so those nutrients have to travel from the land into our water and into our streams to cause these downstream effects. And so I mainly study the processes that control water quality kind of on the front end, where we think about how solutes are produced in terms of what chemical species are generated in a catchment and where they come from, and then how those species are then transported to our streams through different transport processes, and how they're carried through a catchment into our water bodies. And so just to kind of walk through how we think about this, very high level, starting with production, we know that solutes can be produced in a catchment through biogeochemical reactions or added via human inputs, like we've talked about already. And some examples relevant to nitrogen pollution for biogeochemical reactions could be things like the decomposition of organic matter, nitrification, and plant uptake, whereas some human inputs that we think about are atmospheric deposition and the application of fertilizers and manure. And so the solutes that are in our catchment can then be transported to our streams through different hydrological flow paths, as I'm showing in this figure. And so some of the main flow paths that we typically think about include surface or overland flow over the land surface and into our streams. We can also have shallow soil water flow in the subsurface, where we have more of those soils. We have organic matter, plant roots, and things like that. And then lastly, we have this deeper groundwater flow path through the deeper part of the subsurface, where we have our rocks, we have weatherable minerals, and we eventually hit bedrock. And so as water flows through each of these different pathways, it interacts with some of those different materials that I just mentioned. So whether that's minerals or soils, organic matter, and those interactions shape the chemistry of that water as it enters our stream. And so as you can imagine, each flow path has this distinct chemical signature that it ultimately carries into the stream, and then they all mix together to form our stream water chemistry. And so this stream chemistry represents like an integrated signal of all those different flow paths and their chemical signatures, as well as all the processes that lead to the development of those chemical signatures. And so since we typically only have measurements of water quality in streams and rivers and not so often in the subsurface, we often have to infer where those solutes are coming from within a catchment. And so that brings me to one of the very broad questions in this field of research, which is what processes control solute production and transport at the catchment scale. And so one method or tool that I use in my own research to help address this question is reactive transport modeling. And so for anyone unfamiliar, reactive transport models are computational models that couple physical, chemical, and biological processes with mathematical equations. And so we can use them to simulate some of these important subsurface processes like biogeochemical reactions and hydrologic flow paths, so that we can understand how they're influencing our stream water chemistry and those stream chemistry dynamics. And so during my PhD at Penn State, I helped to test and develop a reactive transport model called BioRT-HBV, which has a fairly simple structure and requires minimal input data, and is also computationally inexpensive, which is very helpful in a world where we don't always have the data that we need. And so as a quick overview, BioRT-HBV simulates the hydrology, biogeochemical reactions, and transport processes in a catchment. In this model, we get our hydrology based on the overall water balance between input precipitation, output evapotranspiration, and output discharge. And so just to kind of walk through this figure here, water can enter a catchment through precipitation. Some of that water is partitioned to evapotranspiration, or water evaporating from the land or being transpired by plants. And then the rest of that water infiltrates into our catchment subsurface and then can be routed to the stream through one of those different flow paths that I mentioned. So we have shallow flow, or surface flow, shallow flow through the shallow subsurface, and deeper groundwater flow through the deeper subsurface. And so we can implement some of those different biogeochemical reactions that I mentioned into the different subsurface zones that we have in the model, and that will influence the water chemistry that we have in each of our flow paths. And what's really interesting and helpful about this model is that the structure allows us to look at each individual reaction, each flow path, and each zone to see how each of those impact the kind of integrated signal that we see in stream chemistry, as well as all the different processes that lead to our stream chemistry dynamics. And so in my research, I've mainly focused on studying carbon. And so I've applied this model to the W9 catchment at Sleepers River in Vermont to simulate the production and export of different dissolved carbon species. But you could also use this model to study some of the nutrient species that we were talking about today, like nitrogen and phosphorus processes. And so I'm not going to focus or spend too much time on this example since we're not really focused on carbon today. But I just want to show a few slides here to demonstrate the capability of the model and what sort of insight we can gain from using a model like this. And so in this example, we were focused again on dissolved carbon species. And so the two main solutes that we were looking at were dissolved organic carbon, or DOC, and dissolved inorganic carbon, or DIC. And so those are produced by the different reactions that we've implemented in the subsurface zones there. And so basically when we run the model, we have our hydrology happening, we have these reactions occurring, and then the model calculates the concentrations of each of our solutes in each of our different flow paths and in the total stream chemistry. And so we get those concentrations produced from the model results. We also get flow rates for each of our flow paths and in the total stream discharge. And then we also have reaction rates on a daily time step. And so just to demonstrate, again, some of the insight that we can gain from these sort of model results, we can look at the total rates of net production and export for our two solutes here. So this figure shows 2 years of discharge and air temperature data up top, whereas the bottom two panels have 2 years of the model results where we have production rates in gray and we have export rates in black. And so looking first at the production rates in gray, we can see that both DOC and DIC tend to have higher production rates under warmer conditions where we have our higher temperatures, typically in spring and summer in this catchment. But if we look at the export rates in black, we see that the dynamics here mainly match up with the discharge dynamics up top. And so this kind of highlights the different controls of climate through temperature on driving our solute production, whereas hydrology has a more dominant control on our export rates. And so the reason we care about this is that these have implications for how climate change may affect our water quality moving forward. And so again, this study site was in Vermont in the Northeast US, where climate change projections include increased temperatures and increased frequency and intensity of storm events. And so with these model results, we can make some kind of broad inferences that higher temperatures in the future may lead to increased DOC and DIC production, whereas more frequent storm events may lead to more flushing events and higher discharge conditions that lead to more of these kind of higher export rates of DOC and DIC into our streams. And so overall, that would mean that we have higher concentrations of these dissolved carbon species in our catchment and in our streams, as well as higher fluxes of these dissolved carbon species from our catchment downstream. And so that example just demonstrates how we can use reactive transport models to make connections between observable data like stream chemistry, discharge, and temperature, and then the processes that we can't really easily measure or observe, like biogeochemical reactions and subsurface flow paths. But that kind of leads me to a limitation in this general field of research where our understanding of catchments and their processes is inherently tied to the data that we're able to collect and the climatic conditions that they represent. And so with climate change, those conditions are changing, and that challenges our understanding of these systems and how they function as we continue to observe more conditions that we've never really seen before. And so as an example, we know that climate extreme events are projected to increase in intensity and frequency moving forward, but so far they're relatively understudied in terms of how they affect our water quality. And so if any of you might remember that there were these historic storms and floods in Vermont last summer in 2023. Those storms led to 4 of the 5 highest stream flows that were ever recorded at my main study site at Sleepers River. One of them also flooded the capital city of Montpelier. And so these events are really important to study, and we don't generally have a lot of a clear understanding of how they work and how they are affecting our water quality. And so I'm currently working on applying our reactive transport model on an hourly time step to try to get a better understanding of these event scale dynamics and how they're influencing our solute production, export, and fluxes downstream during these storms. And so that brings me to some of the measurement needs and challenges for capturing and observing these climate extremes. where we really need high-frequency measurements that capture these event scale dynamics. And then we also need to integrate those data with reactive transport models to gain a better understanding of the processes that lead to those observations. However, one of the main challenges that we have in studying these types of climate extremes is how difficult and potentially dangerous it can be to go out into the field and take measurements during this time. However, we know that high-frequency sensors and automated sensors or samplers do exist, and they've worked in a good amount of places, but they also need to be robust enough to withstand any potential damage during these events. And I've heard plenty of examples where people lose sensors or other equipment during these types of storms. But this is, I think, a pretty achievable goal, and I would love to hear if anyone has successful experience in monitoring these types of climate extremes or protecting sensors under these conditions. And another quick example that I wanted to go through in terms of changing conditions due to climate change is the increase that we see in rain on snow events as we're having warming winters. And so here I just want to highlight some work from a collaborator of mine, Dr. Aaron Siebold at the Kansas Geological Survey, who showed that these rain on snow events pose an unquantified continental scale risk of high nutrient export during these wintertime conditions. And so in particular, she was looking at the Mississippi River Basin, which is shown in that black outline sort of in the center of the US, which drains about 40% of the contiguous United States, including a lot of agricultural land in the Midwest and the central US. And so these two maps just show on the left whether an area has high frequency of rain on snow events, and on the right if an area has high sources of nutrients like total nitrogen or total phosphorus. And so if we look kind of at the upper Mississippi River Basin there, we see that they have both high pools of nitrogen and phosphorus, of course, on the right, but also high vulnerability to these rain on snow events. And so this is a problem because these events can flush out those nutrients into our streams and contribute to the high nutrient concentrations that lead to hypoxia, eutrophication, and harmful algal blooms. And so these two plots just show mean turbidity versus mean discharge at two different sites along the Mississippi River Basin. And the dashed line in each plot shows how one particular rain on snow event that they were looking at in 2019 led to a disproportionate amount of sediment transport and likely also high nitrogen and phosphorus concentrations as well, as those species are typically co-transported in our streams and rivers. However, we currently lack sufficient data to fully characterize and monitor those different nutrient sources, their pathways, and their wintertime fluxes. And so these figures just highlight how stream nitrate data during that rain on snow event in 2019 that they were looking at was incomplete during that specific period, but also more generally during our wintertime conditions. And so the authors here were able to use turbidity data to get a general sense of nutrient export, but the exact response of nitrogen and phosphorus during this type of event was not able to be assessed. And so that leads me again to some other measurement needs and challenges in terms of studying these specific rain on snow events, but this also applies to other climate extreme events and studying kind of the difference between extreme conditions and non-extreme conditions, where we really need this kind of continuous year-round monitoring of hydrology, soil, and stream conditions at the watershed scale in order to assess the different processes that are happening and how that leads to changes in our stream water quality. And then another challenge here is that traditional sampling methods and sensors typically aren't designed for cold and icy conditions. And so that's typically why we often have gaps in our data for wintertime conditions. And that's really limited our understanding of different processes that happen during the winter, how those events might be influencing our water quality downstream, and how much they might be contributing to these issues like hypoxia, eutrophication, and algal blooms. And so I talked a lot faster than I really meant to, and I'm sorry about that. But to wrap up, we can see that climate extremes and climate change can have important effects on water quality, but we really need to combine, I think, field efforts and modeling efforts as well to study these events in more detail and to kind of get a better understanding of how much they're contributing to changes in stream water quality moving forward. So I'll leave it there, but thanks very much for your attention, and thanks again to the workshop organizers for putting this on.
Hi, I'm Becky Panchineel. I'm at the University of Maryland. And it actually kind of ties between a few of the different talks. One of the things, for example, with the Chesapeake Bay modeling, is there ways to link how much of the changes or the failure of the BMPs are actually tied to these changing climate conditions where we do have these increased rain events that would feed into the empirical component, but might not be being captured in the modeling component?
Okay, this is on. Well, the Bay program will tell you it's 6% attributed to climate change. Whether or not that number reflects reality is a question, but that's what their model says. The CAS model says is the contribution of climate change. But certainly there's opportunities for more empirical studies of the impacts of climate change in that region.
My name's Rod Kiewit. I'm at Caltech. I'm not a climate or environmental science person. I'm a political scientist, but I also grew up in a farming community in Iowa, and that's my perspective here. The way in which farmers think about fertilizer is as a cost of production, and so they would rather not apply nitrogen that's going to run off, okay, into the streams. It does, but they prefer not to have that happen, because that means they're paying for fertilizer that's not being used. And the method in which they prefer to apply nitrogen fertilizer tends to be through the injection of anhydrous ammonia into the ground, which they claim, and I don't know with the science people, they claim that that results, ceteris paribus, in less nitrogen runoff. Although I have to say Bryn Stewart's point that if you get, because they apply that in the fall, the anhydrous ammonia, in the belief it's going to sit there until the spring, at which point the plants take it up. But if you have a lot of rain, that won't happen. But at any rate, the farmers, I think, are always going to be much more amenable to methods that will reduce nitrogen runoff. And the people who are going to be more resistant are your suburbanites who want a really green lawn. And they will, again, it's on a smaller scale than farming, but from what I understand, the really hot spots in terms of dramatic overuse of nitrogen fertilizer are actually suburban lawns, and that's where the behavioral change is needed, in addition to the farmers. Just an observation.
Thanks. Would anyone like to comment on that for our next question?
Yeah, just in terms of the magnitude, it's a big difference. And so I think it really depends on where you are as to which sources, precisely are important. I just came from Nebraska and Kansas. I was doing a soil health testing and sampling there and working on production farmland. And so I think you're absolutely right that farmers, it is a cost, and farmers would like to reduce that nitrogen input to their land. I think a lot of it is a communication question. How do we share the information that we're learning? Like, for example, we're measuring nitrate leaching on production farmland in Oregon and taking that data and showing how much, we measure at a meter, how much of that fertilizer is moving through the soil and being exported to rivers. And showing that data and showing it year after year that this is what we're finding on their farms, I see some wheels turning, I see some thinking, and some, maybe this is something that could result in changes, but I really do think there's a lot of social science that needs to go into our physical science or biogeochemical science basis to help us communicate and help us tell the story so that those nutrient management plans and those nutrient management issues are updated. And the ammonia doesn't volatilize, but it tends to leach when it's applied that way, is my understanding from just talking to somebody about it 2 days ago. So there's a lot of complexity and a lot of system science that goes into understanding the movement of nitrogen off-site.
They claim that the anhydrous is less likely to move than the granular stuff. That's the claim I don't.
That's probably true, I would agree, but first, plants are inefficient, so you're always going to get a certain amount of loss. I mean, if Robert was here, he would say 40% nutrient use efficiency, 30% nutrient use efficiency. So there's still 6 or 7% of that nitrogen available for transport. I guess the second point, much like what you said, is the magnitude of the problem, I guess. Nitrogen's been applied to some of these lands in the Midwest and Chesapeake Bay, certainly for hundreds of years in some cases. So there's a huge quantity of nitrogen and phosphorus built up in these systems that just acts as that legacy source that's really hard to remediate.
Thank you.
Hi, thank you everybody for great presentations. My name is Neil Fromer. I'm with the Resnick Institute here at Caltech. I guess my specific question is for Kelly, and then there's a broader question that's maybe an implication as a result of that. But with the emit and some of the other remote sensing tools that you were talking about, I guess the question is around the sort of sensitivity for detection of those. Obviously, you're using them diagnostically already for large blooms, things that can be seen in a variety of ways. Is there, sort of what are the detectivity limits there, and what does that tell us about how they might be applied in the future? Or is there an opportunity for them to become predictive in looking at algal blooms or other environmental problems? And then the broader question is, if there isn't, sort of what are the opportunities to think about how we can be more predictive about some of the environmental challenges that are coming there?
Great question. So actually, sorry, I just want to clarify. So when you're talking about sensitivities, it not necessarily like instrument sensitivities, but or like the sensitivity of its like some of these applications, you might be familiar.
Yeah, I don't want to get into the details of the detectivity limits
sure.
of the actual detectors. I might want to, but maybe this group doesn't want to.
Yeah.
But just sort of, how they might be applied, sort of what the sensitivities are in terms of the specific applications.
Yeah, no, this is a really good question. One of the really early, shoes I started to wear was with the Surface Biology Geology mission with the collaborations group. It's a group we started maybe 2 years ago. It's to bring in groups that are underrepresented in Earth Science mission planning process for SBG. And we wanted to start that process early to better understand sensitivities and where there are gaps in knowledge with missions and applications. So for something like emit that comes up, where it's supposed to do mineral mapping and critical minerals, there's a lot of sensitivities around what that could look like and extend to, and really a lot of these, especially when it translates to maybe other types of satellite applications that are definitely on the environmental justice front. We want to make sure we're actually talking to communities really early before you're starting to put out products or satellite information out into the world. So I would say a lot of these conversations are, I would say relatively new, and we're working on learning how to do that. And for the Surface Biology Geology mission, for example, today, we have a drought forum where we had a tribal environmental manager who was like, I would like to use satellite data to look at how does droughts and wildfires impact my community. But like there are several gaps in like how I access that data. What is actually going to be useful to me? So one of the activities we're doing is bringing together missions that are working at drought interface fronts and kind of getting them to better communicate what stage things are at and how some of their applications of their work can be useful. So I'm not truthfully, there's so much work to be done in this space around sensitivity, and I think that's why I'm really excited to be here to make sure that we have more discrete focused things that kind of lead to actual actionable information.
Thank you. I think we have time for one more question.
So this is for Zach. Maybe in your presentation you show biofilters. You were saying that they are not effective because there is a lot of work on pushing these nature-based solution based on wood chip biofilters and others. Are they effective at, for example, reducing nitrate? What's your experience?
Great question. So actually, probably a third of my program looks at evaluating those types of practices too. So bioreactors are incredibly effective at reducing nitrogen. They don't touch other nutrients really like phosphorus, but they're very effective at removing nitrogen from a waste stream. But they're very expensive to install, and there's no agronomic benefit too, so that's why you don't get much uptake from producers.
Great. I think that's all we have time for. Thank you so much to the panel.